Thanks so much for joining us this afternoon. I am Rachel Peterson, the Chief of Staff for All of Us. We are so pleased that you've registered to join our 30-minute open session. We actually have over 1,000 people who registered today. We're going to be featuring a presentation by our Chief Executive Officer, Dr. Josh Denny. But first, as people enter the room, I am pleased to share with you a brief video by one of the researchers using our platform.
I think anyone can be a scientist. You just have to be interested and willing to learn and apply it to, the world around you. I was the first high schooler to access the All of Us data set. The NIH actually created a new pathway for students in high school to be able to access the data set, which allowed other high schoolers to access it too, which was really cool. Once you get into the All of Us, they have many different types of data. They have EHR data, survey data, genomics data. The data is in the workbench. It's all in one central location. They had these tutorial videos on, how to use Jupyter Notebook to analyze the data, how to create a cohort with the specific populations that you're interested in. And that was really helpful in getting started for me. We have a draft paper in progress that's being submitted to various journals with the results from our All of Us data to hopefully help inform health policy and other healthcare researchers. That is what has, encouraged me to go into this field of biomedical engineering. And I don't know if I would be doing that if I hadn't had this opportunity. I definitely do think of myself as a scientist.
Thank you so much. All right, just a couple housekeeping items. The attendees' video and audio are disabled, but we please would encourage you, if you have questions or any technical issues, to use the question and answer Q&A box on the bottom bar. We may respond privately to individual questions, and due to high volume, not all questions may be addressed during the session, but to follow up, feel free to include your email address in your question. Thank you. And Dr. Denny.
Thank you. Thank you, everyone, for joining and taking time out of your day to hear about our update on All of Us. And thank you. I can think of no better way of introing than having Sana's voice and talking about her research as a high school student, trying to break into new domains of researchers using the resource, creating science and altering pathways and future careers. I think this slide talks as an overview of how we are trying to, in one slide, go from our participants to accelerate health research, medical breakthroughs, and to enable individualized prevention, treatment, and care for all. Since we launched nationally in 2018, we've enrolled just about 860,000 participants from all 50 states. That picture on the bottom is a map of ZIP3 zip codes. We actually have 98% of all ZIP3 zip codes in the United States. That's the level of granularity that we make available to researchers in the controlled tier of the database. Those 850,000 plus participants, more than 90% of them agree to let us have their electronic health records. We are approaching collection of nearly 500,000 of those electronic health records. They've provided millions of surveys, millions of biosample aliquots. We take all those data into one cloud platform across the different EHR vendors and systems, even participant donated EHRs, harmonize them, provide privacy preserving approaches, and then make them available to researchers. Again, across all 50 states, more than 1,100 institutions, approaching 17,000 researchers that have access. Nearly two-thirds, or over two-thirds, are early career investigators. The median access time from first login to first starting of creating a project and actually doing work is 29 hours. You can complete that process from one of those 1,100 registered institutions, typically about in an hour and a half or 2 hours if you go straight through. And we're seeing publications pick up. Importantly, a differentiator for our program is returning information to participants. We'll talk some more about this in the session at this point. We're approaching 700,000 individual types of results that have been returned to more than 220,000 participants. The most dramatic of these are those that have received nearly 4,000 people potentially life-changing genetic results in one of the 59 genes associated with inherited cancer syndromes, cardiac arrhythmias, and other conditions that are all actionable if you know that. And these are supported also with clinical test results so that they can act on those. And over 100,000 with information around pharmacogenetics, how their body processes one of 50 or so medications. And about 20% of those actually have been exposed to an actionable medication and have an actionable result. Next slide. So as we talk about our researchers, I mentioned we have researchers across all 50 states, many different types of institutions involved as well, not just the largest research institutions, and as you heard from our video, even to high school students. We have really thought about making this a resource that's accessible. It is free to get on and start using the resource, and you can see the different categories of researchers on the right. Next slide. As we talk about getting into it, we really thought about having this platform be one that we hope makes science both faster, cheaper, and also more reproducible. It becomes transparent both to the participants that can see the research projects that have been started, and then researchers can get access to the very projects, data sets, version numbers, and code used by researchers to look at their work. We have a public tier of data where people can explore what conditions are there, what genetic variants are there, what survey answers are there, and see if the sample sizes are adequate to address a question of interest. And then once you get access into the researcher workbench through either the registered tier or the controlled tier, where you have a little bit more granular information, you have the genetic data, you can start to do those projects. A new researcher goes through one of 6 steps to become a researcher that can actually do work. The first is having that institutional access. That's the institution creates an agreement with Vanderbilt University, which holds the Data and Research Center. And then there's a series of steps after that, steps 2 through 5. After that, institutional access can be completed in about an hour and a half. And we find that the median time is about 29 hours. After people get access, there's no IRB approval needed to start a new project. You just create your workplace description. That can be reviewed by our Resource Access Board. There's a certain series of questions you answer. You can get started doing work immediately. And we also, in our centralized cloud environment, allow our ability to audit and review what is done to maintain trust with our participants. Right now, you receive $300 in free credits once you become a researcher to jumpstart your analyses. Next slide. Privacy and security have always been a top priority for us. The data that participants provide are encrypted at rest and in transit. We remove all obvious identifiers from the data that is on the workbench that researchers see. I've said before that as people use the data are all used within our cloud-based researcher environment. We actually store the biosamples and all the other types of data separately, and the biosamples that stored at Mayo Clinic don't have any direct identifiers on them as well. They just have what we could consider anonymous identifiers that are linked to the Vanderbilt data separately stored that has other types of information on it. And then importantly, we have a certificate of confidentiality, which is part of federal law through the 21st Century Cures Act, which provides legal protection to how these data get used. Our standards also, as a researcher comes in, they go through a training process that talks about training generically around human subjects, but also specific to our platform and our agreements. They repeat and give you refreshers each year. There's a questionnaire, a quiz that they take as well. The workplace usage is monitored, and the project descriptions are public that participants can see and the public can see. And of course, as I've mentioned, they analyze this data within our researcher workbench. Next slide. They don't download it into their own environments. So this just shows a sense of our last 2 data releases and our projected data release in 2026. In the reddish color, we have the release that we put out in 2023, and then In February of 2025, we had our most recent release. You can see that release in the dark blue color brought our participants with survey data to 633,000, our genomic data, whole genome sequences to 414,000. And this is what's available now to researchers. Really excitingly in that part, we took our wearable data composed of Fitbit devices from 15,000 to nearly 60,000. That 60,000 represents the largest wearable data set that I know of longitudinal across the world. Many of these individuals have up to a decade of wearable data. We gave out a number of devices that are ongoing collections as well that is linked to healthcare data, genomic data, those sorts of things as well. You can see we're making steady progress in the electronic health record data collection as well. We continue to work on that through many different avenues to bring that data. And also new long read data, things like long read sequence data. And we're looking in 2026 that our total whole genome sequence data will exceed 500,000, that we will continue to increase the other types of data in here, including bringing in other types of wearable data through Apple Health Kit and data from potentially even things like clinical narratives. We are getting clinical narratives from a handful of sites. We are working on the processes by which we will share these in a privacy-preserving way, probably through extracted concepts, and make those kinds of data available. And we also will probably have some of our first multi-omic data set releases, including proteomic data, RNA-seq data, that will probably happen at some point in 2026. The details of the 2026 release, as you might imagine, are not final, but giving you a projection of what is coming in our plans. Next slide. As we think about what kinds of data we have and the, excuse me, the counts of different diseases we have, this just gives you a count of the various disease counts in the current data set that we have. With over 100,000 people with heart disease and obesity, this encompasses many of the top 10 causes of mortality with tens of thousands or hundreds of thousands of types of cases. And in each of these, we have the combinations of survey data, geospatial data, and other components that allow us to really get at what could be some of the drivers and causes of these chronic diseases. We are working to add in environmental data in addition to the biological and lifestyle data that we have. Next slide. One of the new data sets that we released shortly after the last data set that came out was Exploring the Mind. This is a quantitative assessments of cognitive and behavioral aspects in partnership with the National Institute of Mental Health. This is one of many what we call partnered research studies, where we partner with another institute, center, or office across the NIH at this time, and we will be broadening that to have other partnerships as well, where they help develop and lend their expertise and fund a project on our platform and makes it better. These are what you might call brain games, interactive components that test different aspects and provide these quantitative elements. One of the exciting aspects is we found 10 times more task completion in 6 months of this being live than the previous decade of the project being public outside of this. So we generated a cross-correlated data set to all the other data embedded in our program, and we're able to generate the data rapidly. And of course, there'll be much more data coming out from Exploring the Mind with future data sets. Next slide. We also recently released our first draft of what's we're calling All by All. It is a genome by phenome analysis across electronic health record data and other types of data. This data is available to explore in a preliminary form that's been generated on CDR7, the previous data release. We're looking to update that for the next data release and hope to do that sometime this summer. These data tables are available within the control tier for you to query, so you don't have to query against and perform all these analyses yourself. And there's different types of groupings of the data, as well as a website to explore the pre-calculated results that we've generated here. Next slide. And one of the important things with this most recent data release, it's the first time we've included data from self-identified American Indian and Alaskan Native participants. So as we started this project, we have always enrolled from US locations that did not include tribal lands. But as we know, many individuals who may self-identify as American Indian or Alaskan Native would live in US locations and may join the program. And indeed, through different locations, either as direct volunteers or through our healthcare provider organizations, we had individuals who self-identified as American Indian Alaskan Native join the program. The number in this latest release is 26,000 people who either wholly or partially identify as AIAN. We went through a careful process, as this outlines, of talking to tribal nations through actually 3 formal consultations and listening sessions we did in addition, and created policies, added new protections, new review processes for the data, co-creating some of these processes with tribal working groups and other bodies, including the formal consultations, before releasing these data in the most recent data set. With that QR code, you can read a joint statement from the director of the Tribal Health Research Office at the NIH and myself about this process and the excitement we have to really respect tribal sovereignty, protect these data, and make these data available in a thoughtful process to enable science on and greater research in this very important population that has been under-researched in the past. Next slide. There are a number of publications coming out. As you can see here, they're upticking on the graph on the right. This is what we would expect if you look at other resources. At this point, we see 10 to 15 publications or preprints come out each week. And this is just a collection of some of those. Many different types of publications. We're seeing more publications, as you might expect, come out with genetic data. There are a number of publications that don't use genetic data as well, looking at lifestyle factors and disease associations, thinking about drug repurposing using other approaches. We see AI and ML machine learning papers coming out as well, and across a really broad set of diseases. There are more than 17,000 projects that have been created by our 16,000 plus researchers. So they're really covering the whole range of disease categories and other aspects. Next slide. And one of the things we want to do is translate these to our participants as well. So as we see these papers come out, we create researcher highlights and share these with our participants in newsletters. They're available on the website, and you can use this QR code to see what some of these look like. We just want to make sure that's accessible, and really it's one of the more valuable ways to show how participants' valuable contribution, altruistic contribution of their data and time is making a real difference to them and their communities. Next slide. So the other thing is thinking about our impact. We are young. Our impact is accruing. Publications are just one measure of impact, of course, that we presented before. But the real question is, how do those start to impact real clinical care? And so here are just a couple of examples, and they go from stuff that can be used right now to stuff that is portending towards stuff in the future on the right. And so on the left, as we've done genetic testing and returned these results and classified these variants for participants, we have shared those to the public repository ClinVar. In these first couple submissions that we've made, we're number 14 out of more than 3,000 contributors, so in the top 1%, just on those 59 germline and 7 pharmacogenes that we have looked at. 40,000 variants that we've classified, 8,000 have not been classified before, and of course, these are available to any researcher now, or sorry, any clinician that needs to come to ClinVar and has a participant with this variant. This will add to their ability to reuse those data. There are papers coming out in the middle that will have impact on pharmacogenetic results and interpretation of drug-gene interactions and risk profiles. And some of that is updating guidelines, such as the CPIC guidelines for pharmacogenetics, as you know. And then on the right, just some examples of a handful of papers looking at drug repurposing or genetic variation that may portend to how to treat genetic diseases like APOL1-mediated kidney disease. And then activity is as a primary care physician in my past career, knowing that about 8,000 steps per day is a bend in the curve for type 2 diabetes risk, that doubling of step count from 2,000 to 4,000 has a marked difference actually in risk of disease, is important as we think about how we manage our patient population. Next slide. So this is just a quick review of the progress that's happened since the first announcement of this program to our national launch, and trying to get data out fast to participants and researchers, even as we were enrolling the bulk of our participants, and then starting a pediatric pilot in 2024, and to where we are now. But as we think, one of the things I want to address now is think some about the future. And go to the next slide. Our future is really, the vision is the same despite changes, is to really enable better health for all of us. And go to the next slide. This vision and mission, despite different challenges that we may be facing, as I'll talk about in a few minutes, is the same. And so as we've set our 2025 priorities, this is a high-level view of what we've been focusing on this year. The first is fueling discovery for partnered research studies, those partnerships to launch new studies with other institutes, centers, and offices across the NIH, and eventually beyond the NIH, has really been our primary focus this year. And we've had a lot of progress in this front this year, and we expect to launch 2 new studies this year, in addition to the ones we have ongoing that are making significant progress. We want to really unlock breakthroughs with our biospecimen use. We've generated genomic data. We have, on a small numbers of people, generated long-read sequence data, proteomics data, RNA-seq data that will be coming out. We have not used a lot of those biosamples yet, and we really want to focus on using like the serum and plasma samples, cell-free DNA, RNA at a larger scale, and creating some partnerships and projects that will do that. We are working always on our infrastructures. We've done that with our participant platform, with a new participant platform. I'll talk about that in a second. And then we're working on that with our researcher platform. How do we make that better? And of course, I've already talked about the new data release we're working on. We are restarting our clinical DNA testing results in a new process to participants. And we're really in this new world, and with reduced funding, we've focused on ways that we have better ways to keep in touch with participants digitally. Next slide. So these are some of the partnered research studies we have going across the NIH. The first is Nutrition for Precision Health. This has been cataloged by several media outlets. There's one story here from CBS News. There's also in Good Morning America. It is the world's largest precision nutrition study, and it aims to enroll 8,000 people that do stuff at home and do things like contribute microbiome samples, and then subsets of those go into randomized trials with different diets, and some of those will actually be in a facility and have a controlled diet for 2 weeks, I believe at the time is. We just launched one called Eyes on Health with the National Eye Institute, which will generate optical coherence tomography from 5,000 all of us participants at some of our sites, which of course will have all the other types of data to be linked to it as well. And if that goes successful, we hope that we will be able to expand that in the future. We have an ongoing study looking at people who develop type 2 diabetes since they joined the program and donated their biosamples. And so we took those biosamples, and with the involvement of the National Institute for Environmental Health Sciences, have done an exposomic measure of those. We are generating those data. We will be entering and collecting and putting those data in the workbench, and look if we can look at gene by environment risks for type 2 diabetes. And exploring the mind I talked about earlier, we're up to 84,000 people that have contributed that data now, and we look forward to continuing that and hopefully expanding it with partnership with other institutes and centers in the future. Next slide. We have several new ones coming. So one that's exciting is a partnership with the National Human Genome Research Institute and the NIH Clinical Center to do a project called Genotype to Phenotype, where we will be looking at people that have really rare and ultra-rare genetic variants that look like they may have clinical conditions. It will be used to look at small numbers of people that we can diagnose that may have undiagnosed disease, and also discover new genetic diseases. There are many genes out there that we do not understand if they have clinical phenotypes associated with them or not. And our hope is we may be able to use this resource, call people back to the clinical center, do some investigation. And of course, all that kind of work will be returned to the participant. It will be real clinical testing. Next slide. We are launching one with disability, health, and functioning, and this will look at a tested module out there with our 122,000 participants who have already reported that they have some difficulty to get more understanding of their ability in assistive technologies that they use, and correlate, of course, with health and resilience. And then the Whole Person Health Index is a validated measure that's been used that will help us look at resilience and differences between perceived health and measured health outcomes. And we're looking to continue to grow these kinds of programs and allow external investigator access in an open and transparent way starting in 2026. Next slide. We have started a new process for offering DNA results to participants who want them and who are eligible. Our process here is we've already done the largest that we know of, individual return of results from our research results. And now we have started a no-cost effort to offer truly clinical DNA test results on the same genes, and actually a few more pharmacogenes than we had before, expanding this offering to our participants. We've actually already started this process, and we're going to be doing it in a rolling process to keep up with demand. We have about 139,000 participants that we're working on to offer this to them, and it started already, and we'll be doing over the next couple of months. Next slide. We launched a new participant portal, updating our technology to better help us address the needs for partnered research studies, as well as helping us save money. And in this new platform, you can see some examples of it. Where on the right, we're also looking at how we can bring in more types of healthcare data through other EHR linkages, claims data, what we can do with mortality indices, and these. And we have pilots with a number of different components of this now, and look to expand that over the next year to continue to close that gap in the difference between people who want to give us information and for whom we've collected that information. Next slide. And then for those of you who are All of Us participants, and if you're not, I certainly encourage you to consider joining. We have a new participant platform. If you've not logged in the last couple of months, I encourage you to go to the website. You will have to reset your password. You'll click that forgot link there. You'll get an email, reset it. You'll go back in, and you'll be connected to that platform. All your data will be there. Your connections will be there, and you'll be able to start receiving our updates, and as well as if you have any activities to complete, we would love for you to do those with new surveys and that sort of thing. Next slide. In this 2025 year, we also look to start our first reassessment. This will be a digital reassessment set of surveys that will be offered that will borrow from the existing survey data, physical measures that we have done before. We will not be collecting biosamples with this first reassessment. It will be a digital one. We hope to do biosample recollections in the future. And of course, I already mentioned some of the other future surveys that we have going on. The EHR data, other types of data like Fitbit data, continue to accrue longitudinally. Next slide. And finally, I want to talk a little bit about one of the biggest things we have in our future environment, which is pediatrics. We did a successful pilot. As we changed participant platforms, we have rebuilt the technology, and we've relaunched pediatric enrollment for 0 to 4. We are working on protocols and technology for older age groups. And, this was a pilot across 5 sites. We believe we have the technology. We have a number of adult participants who have children that we know would be interested in enrolling their children. And doing so would really help us cross the lifespan, truly be a program that can cross from birth to death, and capture if we do this in a large way, as funding permits, we would be able to look at those earliest life involvement of things that affect our health longitudinally. Next slide. One of the things I've alluded to a couple of times over the last 2 years, due to decreases in blue sorry, I'll get this right, in orange of what was 21st Century Cures funding established in 2016. We have seen a budget reduction of 34% last year and a total of 71% this year. It's forced us to do some really strict make really strict decisions in how we prioritize where we spend our funding. It's caused us to dramatically change and reduce the number of adult enrollment sites, a lot of our engagement partners, and functionally will be pausing most adult biosample collection. We are keeping the digital front door open for enrollment, and it's the major reason why we have not launched pediatrics in a large way at this point. We have focused on this time in ways that we can cut costs, and we've saved really tens of millions of dollars in prospective costs in how we have consolidated with technology partners and changed some other infrastructure pieces, and even how we've optimized our DNA return to results strategy to provide both greater value but also reduce cost. But during this time, we have grown our collaborations across the NIH to be a platform to launch new research studies, both using the researcher workbench, but the partnered research studies, which continue to grow. And we have 18 additional partner research studies in progress in discussion points in addition to the ones that have already launched. So that is a fruitful pipeline that we think will continue to grow the resource. Next slide. And as we think about us, I really want to position all of us as a health research platform for America and through broad access for the world. I think about this in terms of our impact, and we really want to see ourselves progressively move towards clinical impact. That will take more time to accrue, but we're already seeing some of it through things like ClinVar sharing of genetic variants, some discoveries that are leading towards actionability and point towards potential new therapies. We're trying to make the platform as efficient as possible and how we can both make science happen faster in a more reproducible, transparent way, as well as be cost effective by centralizing resources, allowing people to share data and start new studies much cheaper than they could have otherwise. And innovation is key to this, and how we think about this and how we really allow ourselves to be a platform that many people use, and they generate the science on top of it, and how do we support that in flexible ways that, of course, preserve our protections for our participants. Next slide. And if you're not receiving communications from us, I encourage you to subscribe to things like our newsletters. If you're a participant, make sure you have your settings to receive these kinds of communications. We're doing regional-based participant convenings across the country. We had one last night with our Southeastern participants, and we will continue to do those across the country this year, which is exciting. And then periodic updates like this. I will continue to come to you with open sessions to tell you updates and what we're planning for the program. Next slide. And with that, I just want to thank you for your time and happy to take a few audience questions.
Thank you so much, Dr. Denny. I do have some wonderful questions we received in the Q&A. I will be fast if you will be fast. I know we're a couple minutes over time. First one, will participants be able to receive their sequences back to them individually?
The being able to receive a raw sequence result is something from our research data that has been governed by the FDA, and we do not have approval to do that at this point. I would love to be able to do that, and it is something we are exploring as a future kind of result. We just don't have the ability to do that right now. What we are continuing and will to offer back health-related genetic results, and the results we're returning also include things like ancestry and traits information with that. And of course, that includes the actionable variants that are part of that.
Thank you so much. With the privacy rule of 20 count, limiting publications on rare variants, how is the program balancing privacy protections with the scientific utility of the data, especially for rare disease research?
Yeah. So one thing I want to point out, rare diseases can get into very low sample size counts. And we have a rule that governs researchers as they make publications that if any cell counts are less than 20, it's not that they can't share it, but they have to ask us for our approval first. And then we evaluate the privacy implications of sharing that data versus the scientific benefit. And so a number of papers have asked for that approval and shared really important information on very low sample size counts for rare genetic diseases or other kinds of things like that that require those kinds of data. And we always do a balancing act of thinking about how do we make sure we're protecting privacy, but let the important science that requires that information to go forward, to be released, to go forward in a safe way. And so we're always doing that balancing, and we have a mixed committee that includes participants that looks at those requests.
Thank you so much. There are so many wonderful questions. I'm just going to leave us with one more, which is, when do you expect the data from Nutrition for Precision Health to be added to the researcher workbench? And is there a repeatable framework in place or a mandate for partnered research studies to share back that data collected from the participants?
Great questions. The second question is the easy one, which is yes, those data are any data from a partnered research study will be shared through the researcher workbench to broad researchers. And as we develop these partnered research studies, we're actually looking also at what are our opportunities as we generate data to return data to participants as well. So Nutrition for Precision Health is in an ongoing way sharing different types of information with participants that are part of that study. For example, with the eye imaging study, we share a picture of, we share some of the retinal imaging as photographs that they can take with them, and then take to their doctor if they want, or whatever use they might have for them. In terms of the data set release for Nutrition for Precision Health, I don't actually recall the timeline for that release. And so in order to not be wrong, I'm not going to give an answer for that. I know that is still actively enrolling, and what's one thing to point out is when you're dealing with something that has a clinical trial attached to it, you have to reach certain counts before you'd actually release the data. So that's going to be one of the things that moderates when the NPH data actually goes into the workbench.
Thank you so much. And with that, we will go ahead and conclude our open session. We appreciate you all joining us.